All right, ladies and gents, welcome. Today we're going to talk about how your cells are able to get nutrients in and get waste products out. Last unit we were talking about your cell structure, so things like the Golgi bodies, the mitochondria, the chloroplast, the nucleus. But in order for any of those organelles to be able to do their job, they need to be able to get materials out of their environment and then get rid of the waste products that they create. And in order to do all of that, that's what we're talking about this unit as far as cell transport. Now, to get anything in and out of your cells, that means we're going to have to cross the cell's major barrier, which is your cell membrane. And so your cells don't have a brain. There aren't little aliens living in your cell membrane to be able to decide what's capable of coming into the cell and what's capable of leaving. What sort of things do I want to get into my cell, and what are the things that I need to get out? Because I don't necessarily want everything in my environment to be filtering into my cytoplasm. And the reverse is also true. Things that I have in my cytoplasm inside my cell, I don't necessarily want all of that to be able to leave. And so it's really going to come back to your biochemistry. The major structure of your cell membrane is these phospholipids. Remember that they form that double layer, that bilayer that surrounds your cell membrane. And it really is going to come down to the chemistry of this particular macromolecule that dictates how certain things are able to come through and why not everything can pass. So if we remember, your phospholipids are amphipathic molecules, meaning they have part of the molecule that's hydrophilic, it likes water, while other parts of it do not. So when I look at the head of the molecule, this is the part that's hydrophilic. This is the part that likes to be connected to water because it's polar. That phosphate group in there, those charges that you're seeing in the picture, are what makes it polar. So those charges are able to interact with the partial charges on water, hence why it likes to interact, why it's hydrophilic. On the other side are your tails, and these guys are hydrophobic. They're just big hydrocarbon chains, carbons and hydrogens linked together. And as a result of that, they are nonpolar. And so they are not like water. There are no charges here for water to interact with. And as a result, it's like oil. So they aren't going to interact at all. It's kind of going to sit there and get separated. So your cell membrane organizes itself into this bilayer so that those hydrophilic polar heads can sit on the outside of your cell membrane and interact with the water on the outside of the cell, all right, as well as interacting with the water in your cytoplasm. Remember, what your cytoplasm is mostly water. It's that cytosol, all right, and so there's a copious amount of water inside. By layering themselves or sandwiching themselves into this bilayer, hydrophilic heads can interact with the water in and outside of the cell's environment, and those hydrophobic tails end up in the center and are basically shielded by those heads from any water source. So this creates that boundary between what's outside your cell and inside, and this is a really big evolutionary step as we started to organize into these cellular structures, because now I can keep certain things in, I can get chemical reactions to happen inside my cells in a much more closed environment, protected environment, so they're more efficient, and I can keep things out of my cells that I don't want to interfere with those sorts of chemical processes. So what this ends up forming is this selectively permeable barrier. Now let's break that word down. Selective in the sense that some things can go through, some things won't. All right, and it's permeable in that permeability means that things can pass. All right, but because it's selectively permeable, it's going to control the flow of substances. Some things can go in, some things can go out, and I can control what that is and when that happens. And all of this is going to come down to your cell's ability to maintain homeostasis, to be able to keep these steady internal conditions inside my cell, regardless or in competition with whatever's going on outside in its environment. So what's going to really affect the kinds of things that are capable of crossing those membranes? Well, it comes back to your chemistry. So when I'm looking at certain molecules that are going to pass across that membrane, one of the things I'm going to have to take into consideration is size. The space in between those phospholipids is only so big. There's some flexibility, there's some movement there, but I don't want to be trying to force some giant molecule through that membrane. It's just going to blow a big hole in it and kind of defeat its purpose as a whole. So size is going to make a difference. Shape may also make a difference, and we'll see that when we talk a little bit about some of the other structures that are part of your cell membrane. And the other big part is the type of molecules we're talking about. So when we classify molecules across the board, they either fit into that ionic compound idea. So when it dissolves in water, the ions come apart. So either they're positive or negative ions. They've got a full charge. They can be hydrophobic molecules, which is where the nonpolar stuff comes in, right? Or they can be hydrophilic, and then they're polar, so they have partial charges. All right, so really we're looking at, do I have some sort of charge, whether it's full like an ion or partial like a polar molecule, or do I have no charge at all, in which case I'd fall into that nonpolar idea. And so it's the chemical makeup of those molecules that I'm trying to send through the membrane, as well as the chemistry of those phospholipids as a whole, that makes this membrane selectively permeable. It has sort of, quote unquote, this ability to choose what goes through and what doesn't based on whether or not the molecules I'm trying to get through the membrane are able to interact with those fatty acid tails. So remember this whole idea that like dissolves like. If I'm going to interact with another molecule, it needs to be the same chemistry as myself. So when I'm looking at my cell membrane structure, it really is going to be this nonpolar tail hydrophobic layer that really is going to be the barrier. All right, can I get through these heads, and then can I interact here in order to pass the rest of the way through? So anything that's small and anything that's nonpolar, so size also matters. Anything small and nonpolar is going to be able to go directly through this membrane, no problem. All right, so things like your gases, oxygen, carbon dioxide, those sorts of things are going to pass straight through. No need for anything extra to help. On the flip side to that, though, anything that's big, and or anything that has some sort of charge, whether it's an ion or something polar, is going to need help getting through. And for most of those molecules, it's going to be in the form of some sort of protein. Think about even water. We know that your cells need water in order to survive. Heck, the vast majority of its internal environment is water, but water is a polar molecule. So in order for water to get through this membrane, whether it's coming in as some sort of nutrient or going out as some sort of waste, I'm going to actually need some extra help. I'm going to need some proteins to help me get through that membrane because they're not going to be able to interact with those nonpolar tails. Now, when we look at your cell membrane, this picture is much more representative of what a full membrane structure looks like. This is a much more watered down version. All right, so what else do we have going on here? Well, it's really this idea of this fluid mosaic model is a way that scientists use to describe what your cell membrane structure truly looks like. So we've got that phospholipid bilayer. The vast majority of that structure is those phospholipids. But if you take a look, there's a lot more complexity to this membrane structure than just a bunch of phospholipids. Okay. There are all sorts of different proteins, and we're going to spend a couple of minutes here talking about what those proteins may or may not be doing in terms of function, because they will have different functions. Other ones that are important are things like these carbohydrates that are sticking off the exterior of that cell membrane, and most of those carbohydrates are used for identification tags. Remember that especially in multicellular organisms, your cells don't have brains, they don't have mouths, they don't have eyes, they don't have ears. There's no way for them to communicate in a sense that we typically think of human communication, right? Being able to see people's body language, hearing their tone of voice. Your cells can't do that, but at the same time, they still need to be able to communicate and coordinate their efforts. So, one way that your body is able to identify what kind of cell it's dealing with are these carbohydrates. It's able to be used to identify that, yes, this cell actually belongs to me, as opposed to, say, a pathogen, a bacteria, or a virus that might be invading, and we need to take care of that problem. And it's also a way for your body to identify all these different types of cells that you have in your body. How do I know that this is a muscle cell versus a neuron versus a red blood cell? Well, these carbohydrates that are sticking off of your cells are a way for your cells to be able to identify themselves. You've also got cholesterol that's built into that membrane structure as well. And remember that cholesterol tends to get a bad rap, right? We talk about high cholesterol and cardiovascular disease and heart attacks. That's true. But when we actually talk about cholesterol from a health standpoint, there's really two types. You've got your HDLs and your LDLs. One's good, one's bad. You do need some cholesterol in your body, and that good cholesterol needs to be here in your cell membrane structure because it's going to act as a temperature buffer. All right. So when your cells get in a situation where they're sort of below typical normal temperatures, all right, within some reasonable level, this cholesterol can help keep the fluidity of that membrane, keep it fluid, keep it moving, being able to allow some space to form so it can pass oxygen or carbon dioxide through it. The same goes on the other end. If I'm getting above my normal temperature level, again, within a reasonable range, that cholesterol can help to sort of solidify and keep that membrane solid without you all just kind of having your membranes flowing all over the place and everything kind of breaking down. So all of this structure here is there to maintain the protection of what's inside your cells from the external environment and be able to monitor and maintain that homeostasis. So I mentioned to you on the previous slide that there are a lot of different proteins that are sitting in your cell membrane, and they do a whole range of functions. One type of function is to go back to that communication piece, right? Your cells need to be able to talk to each other, and all of that quote unquote talking really is coming down to molecules chemistry. So some of these proteins act as receptors. They receive messages in the form of actual protein messenger molecules or some other type of molecule here. That messenger attaches to a very specifically shaped protein. So go back to that shape idea that we talked about before. So they'll kind of like lock and key, like an enzyme. They'll only attach to the receptors that they're meant to, and that will then trigger a response inside my cells. So that receptor protein is being detected by this receptor. It's like getting a signal from a satellite onto your cell phone. That signal specific to your cell phone number, and then you pick it up, and it's going to cause you to do some sort of response. Say hello. It's your mom on the other side saying, "Hey, can you pick up milk on your way home from school?" That kind of thing. But what do your cells need to do? So that activated molecule may go trigger some chemical reaction in your cells. It may go trigger your cell to start making more cells. It really kind of just depends on what that signal is. And your cells have very specific receptors connected to very specifically shaped messenger molecules so that it can regulate what task gets activated once that recepted message comes in. Some of those receptor proteins may actually function in terms of cell transport. So some of those receptors will actually change the permeability of the membrane. So this is one way for me to control what goes in and what goes out. So I've got these proteins here, these receptor proteins that are connected to channels. All right. Kind of think of them like gates. So if that gate is closed because no message has come in to tell me I need to move these materials in and out, then the gate stays closed. In comes that messenger molecule, connects to that ion receptor, and basically opens the gate. So now I can allow certain things to go through. This is only going to work though for molecules like sodium, potassium, chloride, ions, or any other polar molecule. Because remember, anything nonpolar can go straight through those tails. All right, but this gives me some more control over moving anything that's ionic or polar. Some of those receptor proteins can also act as second messengers. All right, and what does that mean? It's just a more complex pathway. So in comes that receptor, it's going to attract another protein that changes shape, that changes some effector protein like an enzyme, that then activates some more molecules and causes this cascade of chemical reactions in your cells. All right. And those can do similar things to the channel proteins we just saw. Um, all this does is allow your cells to have some more control over those multiple steps, right? So rather than it just being sort of on or off, like this receptor protein here, this now gives me multiple steps of control. All right, does make the system a little more complicated, but allows me to turn on and turn off with more of a fine-tuned knob, if you want to think of it that way. So rather than just being kind of an on-off switch like this, now I've kind of got a dial. I can tone it up, I can turn it down, so there's some more fine-tuned control here. Other receptor proteins may act just like enzymes, so once again, triggering enzymatic reactions, speeding up chemical reactions in your cells by allowing these guys to be triggered by some sort of substrate that comes in. And then what we're going to focus on mostly for this unit are those transport proteins. Now, some of them can be like those gated channels we just saw where the receptor comes in and opens them up, keeps them closed when we don't want them. Others will just be channels that are open all the time. Something like an aquaporin, aqua for water. Water pores is literally what that translates to. Remember, water's polar, so it can't go through these tails, but I constantly need a flow of water in and out. So those aquaporins are open channels all the time. They don't require any energy to use them. They just work based off of concentration gradients, and we'll get into more of what that means as we move forward. Others, however, are going to act like pump systems, in which case we're going to need energy. Remember, ATP is our big energy molecule in our cells, so I'm going to need to spend some energy to move things, pump things, just like electricity would need to manage a pump in your house, say in your basement if you've got a sump pump to pump out excess water. Okay, so some of them will require that energy and some of them won't. It kind of just depends on what their function is.